Testing, Ken.
I'm on mute. Yes, there you go. Hello, can you hear me? People on chat, can you press the some button or something? There you go. Okay. So as I said, as Luzine said, I'm a postdoc in Lab, and I've been working on the Lightsheet microscope for a good amount of time. I've been clearing a lot of brains for the past couple of years. And for me, the biggest struggle was actually this pre-processing part. Thanks to everybody in the field and thanks to Life Canvas, the clearing is so easy, actually. You just like put the brains into solutions and you get the brain out. It gets remarkably transparent. You put it on the microscope, you get like fantastic images. But from there, to convert that into something that's meaningful is actually a struggle. The reason is that you have like 2,000 images per channel. That gets you like 80 to 100 gigabytes. And then you have multiple channels, and then you have like 10 animals, 20 animals, and then you end up like 100, 500 gigabytes and like terabytes of data. What's also challenging is that it's not like you can like parse out one Z slice and do something like that. You have to handle it as a whole. It requires a lot of memory on your machine. It's not like you can just put it on ImageJ and check it. You can't even do that if you don't have a good machine. So the challenge to extract something out of that very massive data is going to be what we're going to be talking about today. It requires certain understanding of efficient computation and image processing. In also some cases, you need some knowledge about specific hardwares. So the purpose of this portion of the workshop is to offer you like as a simplest pipeline as possible that you can just like put in your data, change some variables, and at least get something out of it. Okay. We hope that like you don't have to worry about too much of setting up the system, and you can just directly go into the data and like directly go into like troubleshooting, at least troubleshooting to like the quality of the data part. Also, I wanted to stress out this is that not because Chris and Live Canvas is here, there's actually services that you can outsource this pre-processing pipeline. I think, are you going to talk about that a bit? Okay, but like there is gonna be some outsourcing resources where you can just hand data to somebody and get the data in. I highly recommend, even if you will be doing that, and I think that's actually a great idea. It's a balance of like price and like time. But even if you're going to do that, knowing how it is going to go through, what kind of steps it's going to go through is very critical. So you can like do some more efficient quality control of what you get from those companies.
Yeah, we do have some customers who will make a decision based on the project size, like they'll have some of those in-house, and they will say if a product size gets to a certain number, they're computation constrained. And they actually outsource those types of projects to us to do them, and they do the smaller types of projects in-house.
So for me, I did go through some of those processes, and then the decision I made is that I wanted to have more handle among how the data is processed. I wanted to adjust some variables. I wanted to do some more adjustments. So that was the reason I went through all this process to like be able to do it on myself. But there is always an option if you already know what you are going to get and you just want to speed up the process, of course, then outsourcing is a good idea. Okay, so that being said, let's go in. So the obvious question is, so what are we going to do today? The first half will be just talking about what is pre-processing and what we want to do. In the latter half, we'll do some hands-on demo and we'll ask you and have you guys handle some actual data, process that on a cluster computer. Okay. So the very first question is very simple. What is pre-processing? I've talked a bit about this a little bit, but pre-processing is a necessary for any kind of experiment where the acquired data does not actually provide you the quantified results. The raw data you extract out of an experiment is not necessarily in the form that makes sense to you. Like, for example, for fiber photometry data, the raw data is just a time series of photosensors signals. It's just like a fluctuation of like a specific value. You need to correct for motions and like what you have to align it to behavior data, and then you can extract something out of it. For our things, so for Lightsheet data, the raw data, as I said, is images, but the type of data you want to extract is somewhat dependent on your experiment. But it's very extensive, and you have to have some quality control for each step. For like very popular examples, if you want to quantify the CFOS positive cells in the mouse brain, you start from stitching to create the image per Z-plane. You filter the image so that you can just remove some artifact signals. You have to resample the image because what you want to align the image is onto a brain atlas, and that is in a completely different resolution. So you want to resample, reorient to so like it kind of aligns, and then you have the actual alignment process where you
bugs you just let me know. No problem. Okay.
where you try to somebody's talking. Okay. But you basically want to just align the two images. Then you have the part of segmentation where you actually try to segment the cells out of the data. You transform the position of the cells into raw atlas space and to the atlas space, and then you quantify the cells per region or do some voxelization to make to quantify the distribution of cells in space. So even to get out the results you want to get, like such as cells per regions or like voxelized data, there's like this many steps you have to go through. This changes if you want to do something else. If you want to quantify YFP axons in a rat brain, there's a couple of steps that's similar. You have to do the stitching, you have to do the filtering, you have to do resampling, but you have to change the variables depending on some things. For example, one is that you're not going to go for the mouse atlas, you're going to go for the rat atlas. So you have to like modify your pipeline so that it works well with the rat atlas. You have to like provide the pipeline with the rat atlas data. You can't just segment a cell, you have to segment axons. And then for when you segment axons, you do steps like skeletonization in order to like just normalize the like a width of axons to some template. And then you transform not the positions of the axon, positions of the cells, but you transform the axons mask onto the atlas so you can quantify the entire area of the axon or like the intensity of axon or whatever. So these are two very extreme examples, but what you need for each step is very different. And then you have to choose what you need to do, and you have to have, again, the quality control checks in each point. Okay, so obviously the last part of this is because of the data is massive, you just can't put them into like ImageJ and manually quantify them. You have to have some kind of automatic process in the way. So, and also you have to make this reproducible. So you have to have a very consistent code that goes through everything in the same manner. So that's why you need to have some understanding of like what kind of packages, what kind of codes that you're going to use to process all these data. Okay. So what do you need for pre-processing? I think there's basically three things. One is obviously in terms of softwares or like data, there's basically three things. One thing you need is a template atlas files, which is what you want to align your data, your brain data onto. The other thing you need is some very critical pre-processing packages. We will be using a package called ClearMap in this workshop. ClearMap is a Python package that covers resampling, alignment, segmentation, transformation, quantification of cell type objects. It's a type of package that's kind of a wrapper. So it has very different like modules or functions in one big package. You use like the resampling function or the resampling module in this package to do that and that and that. So it's a very like well designed, well organized package. One thing to keep in mind that these packages are just like growing and being developed day by day. So there could be something better for you. But for today, we're just gonna stick on ClearMap because it's just well designed and we're used to it. So other related environment files is something that we will typically install in a computer or set up your computer so that the pre-processing package will work on. People, if you are familiar with like Conda, those virtual environment things, that's what I'm kind of referring to. So you also need that kind of environment so that your like key package will be working on. So the first thing we need to talk about is these template atlas files. I'm listing 3 examples of the brain atlas that we have experience using it within the NAPE Center. So the most common one will be this Allen CCFV3. So this was obviously developed by the Allen Atlas. I don't think I need to give you any explanation about this, but like the Allen Atlas has used like 1,675 young adult B6J mouse brains to create this like template MRI image, and they have like data-driven or organized segmentation of the brain regions. You can like look it up on their GUI on their webpage. It's very commonly used to identify regions in the mouse brain. The second one is called the Kim Unified Anatomical Atlas. This was developed by Yoo-Sung Kim. The intention of this atlas was to make it a little bit more closer to the traditionally used Franklin and Paxino's mouse brain atlas. One thing that a lot of people had concerns with the Allen atlas was some of the subcortical regions, especially in the hypothalamus or like the amygdala, were not well segmented or not well defined as we traditionally understood those regions. So what Kim Unified Anatomical Atlas tried to do was to use some of the Allen CCF framework, but also integrate the Franklin Paxino's brain region. So this is actually more commonly used in the NAPE Center right now. I have been using this one because it had very well defined hypothalamic regions, which I was interested in. But the point I want to convey here is that depending on what you want to study, you really have to you can actually select what kind of brain atlas you want to use. And if one brain atlas suits you better, you should use that. I think the biggest merit of using Allen is because everybody is using it. You can integrate other types of data that is already mapped onto the Allen atlas and compare it with your results. The third one is a bit different. This is the rat brain atlas. I think Cassidy has been working on this type of atlas, and I don't know how many rat people there is today, but having a good like rat brain atlas in this structure has been very important for people in rats to use Lightsheet brains, Lightsheet microscopy. This I think is the current maybe the best standard at least. There's 222 structures with 112 new 57 revised regions, I guess. I think some people have some concerns about this atlas. Typically, some regions are more well-defined than others. There's some kind of like, there are pros and cons. But there is, at least we can say that there is an atlas if you want to use it as your template. Okay. So this is what the atlas is looking like as an overview. But when you look at like what kind of files do you actually need, there's actually 3 types of files. One type of file is this atlas file, which is an image file that will resemble your background image. So this is, for the outline, this was an MRI image composed from like 1,600 mouse brains. This is what your brain will actually be aligned to. You can see that there's like a lot of like landmarks, like this hippocampal structure or like this like commissures and ventricles and stuff like that. So these key landmarks should be present in your autofluorescence image as well, because the elastics transformation algorithm will be using these landmarks to align your brain atlas, your brain images onto the atlas. The second file will be this annotation file. So this will be an image file that will be in the same shape as this atlas file. So it's the exact same shape, but it doesn't have this like raw autofluorescence-ish signals. It has this like different colors for different regions. So what it actually means is that each region that is different in color has a specific value filled inside that region. The third file, this atlas info file, is something that we'll be using to decode that. What this means is that each region will have an ID. And because an atlas will form like a tree type hierarchy structure, on the top, there will be like the entire brain, there will be the gray, there will be the cerebellum, and then the cortex and so on. So it's like a tree structure. You go from the very broader region to the very finer regions. This atlas info file is used to construct that structure by assigning a parent ID for each region. So the way it works is that, for example, primary motor cortex layer 1 will have a parent region of the motor cortex itself, or the motor cortex 1 itself. So like you have this region, you have this parent region, that region will also have a parent region, and so on, and you can reconstruct the entire tree. Each region will have an ID, and then there will also be a parent ID. The way it works is that if you look, like if you take this annotation image in image J and point to a specific position in this space, you will get this value of 943. This 943 will be corresponding to the primary motor cortex layer 2 free. So you can see, okay, this region is primary motor cortex layer 2 free, right? So if you understand this, these 3 images, these 3 files are what you need to provide to the ClearMap pipeline in order to like, we call it transferring labels from the atlas file to your actual image. Okay, so the next thing we will go into is what is ClearMap. So as I said, ClearMap is a toolbox for the analysis and registration of the volumetric data from clear tissue. It was published at least in 2016. It came with a data set doing CFOS staining in the entire brain. So it was originally made to quantify CFOS positive cells in the brain. And it also came with this alignment and resampling functions. It's very close to what we do. So like, it's really easy for us to like understand and use. It runs on Python 2. Which is a kind of like a older type of Python. So this is one reason that we really have to care about like the environment structure. So like we just can't like install it in any kind of computer, which typically runs in Python 3 nowadays. So the outline of ClearMap preprocessed pipeline will look like this. So you will start from these type of data. You will have the Lightsheet, the images that came out from Lightsheet microscope. You will have a signal channel images and then all the fluorescence images. And you will have these template atlas files that I was just talking about. The ClearMap can do first, you can do the cell detection. So it will use all the signal channel images because there is where the signals are. It goes through a function called the detection function. It will give you using certain types of algorithms that I will be talking later to identify the center of the cells, and it will give you those cell coordinates as well as the cell intensity and the cell size for each of those masks. You can also apply a filtering, which means that you can threshold the cells by size or intensity. You can like omit all the cells that are bigger than whatever, smaller than whatever. The other thing you will do in parallel will be the alignment. So to alignment, you have like these three lines, but basically what you're trying to do is align these two images onto the Alice file. You would suspect that you might think that you only need the autofluorescence image file to align through the Alice file. But in reality, there are cases where the signal channel image is a bit offset from the autofluorescence images. So what it actually does, it aligns the signal images onto the autofluorescence images, as well as aligning autofluorescence images into the Alice images. So this is through the alignment process. And what you will get as a result is what we call a transformation vectors. So these vectors will be unique to and will allow you to transform a certain image or a certain shaped array into a different type of array. So once you get this, you can transform like any sort of things that's inside here into this thing, or like the vice versa. Okay, so next thing will be to applying the cell coordinates and transforming into outer space using the transformation vectors. So because you have these cell coordinates, you know where it is in like the raw image space. You will be using the transformation vectors to convert that in the positions in the outer space. This will be again done inside a function called the transformation function. Okay. The voxelization is something a bit something that we will be talking later this afternoon, but a way to visualize the positions of the cells in a mapped Alice space. Because there's so many small dots in the brain that there's so many CFOS cells in the raw image space. If you convert that into the Alice space, it will be like super tiny dots all over the place. Voxelization is just a way to like more like round it up and visualize that in 3D space, in the Alice space. Okay. So after this, there's also one more step. It's a kind of a label transfer and quantification step. Because you now know the cell coordinate in Alice space, you can use that information together with the annotation file and the Alice info file to say, okay, this cell is in coordinate this, so it is in this region. So you can count, okay, one cell in this region. You do that for all the cell coordinates, and at the very end, what you will get is that you have like 10 cells in region 145, 100 regions in region like 764, or something like that. You convert that IDs into region names, or like Cortex layer 5 or something like that, using the Alice info file. And as a result, you will get the cell count per region data. Okay, is that clear? Okay. The type of images are looking like this. You start with like TIFF series images, or like TIFF images here. All these cell intensity or cell coordinates are NumPy arrays. Transformation vectors are basically text files, and NumPy arrays, TIFF image, and CSV files, so on. The final data that you will be using for your analysis will mainly be these TIFF images and CSV files. So again, you start from like this humongous data set, but what you actually get is like 100 or 200 megabyte TIFF image and like a 200 kilobyte CSV file actually. Okay. So these are like the main ClearMap functions. I think I went through most of them like already. I will be going through them in more detail when we start like looking at the code in the hands-on session. There's a good document from the developer of ClearMap in this GitHub webpage. And when you can use that as a reference, they have a good tutorial too. Okay. So now I'm going to talk about a little bit of like quality control checkpoint steps. The first thing that you will have to think about when like starting to analyze your data using ClearMap is you want to get the cell detection step very, very good, because that's obviously your key step if you want to do like CFOS cell counting. ClearMap provides you with the tools, but you still need to do some optimization in order to like get the spot detection work very well. ClearMap offers two segmentation algorithms or segmentation approaches. One is called the spot detection approaches, which applies very simple filters onto the image and does a maximum detection. It's a very simple way to quantify a peak of intensity in 3D space. The elastic detection, which we'll be talking about later today, is using a machine learning algorithm called a machine learning classifier called that is trained in elastic and use that to classify cell pixels. In either way, spot detection is more simple. As I said, it works really well in like nuclear signals, very round signals that doesn't have a lot of like weird shapes. It works well on those. Elastic detection works very well in like other types of images. You can like train your classifier as you want. So like it gives you a lot of more like room to modify your segmentation quality. It's a
So if anybody's looking to like segment cells, or I seem like tracing or cells in general, spot detection for us in the Golden Lab was only useful for whole grain resolution data sets of like imaging at like 2x, so like above 6 micron isotropic resolution. Anything like higher than that, or lower than that. So anything higher than that, spot detection didn't work very well. So we had to stick to classification. So if you're looking to do, if you're imaging at like 1.6x on the smart spim, which I think a lot of people will be doing, and if you want to count cells from like CFOS or AI14, that is labeling where your trap creator line is labeling, then I would you're probably going to want to use elastic just to let you know. But if you're imaging at a higher resolution, you want to count cells in a smaller data set, you might be able to do something. I don't know what your experience.
No, I've been using 1.6x for all my 1.6x CFOS data is all done in spot detection.
You're doing both.
Yes, I think it will work fine. So this is my work case. So for nuclear signals, this is a CFOS stain image. This I will be using spot detection. The data set that you will be handling this later today will also be these type of images. We will go through a spot detection. These is coming from a traps animal. So this is Tito tomato filled cells. This I will be using elastic. These are both imaged in 1.6x objective, but you see like some of these dendrites really mess up with the segmentation. It is like artificially, especially when there's a lot of dendrites very dense, it will form this like very strong signal and be artificially found as a cell kind of object. Okay, so I would recommend, and we will also go through this during the hands-on part. I typically recommend to create 3 to 10 images that are roughly again 100 and 100 and 100 pixel size, and manually label the cells if you are first training a classifier. You can do that with ImageJ or whatever, but the key here is that you know what the true cell locations are. And then you can compare the results coming from ClearMap cell detection and to your manual labels. And then you can quantify, you can also like quantify like things like the accuracy or precision or the F1 scores of how well the classifier or like the segmentation variables worked for your data set. This is a very important part because, and you should use a lot of your time to do this before you like apply your segmentation filters, segmentation classifiers on your full data. Because again, you know, working on the full data is just a kind of a black box. You really need to know that it will work in small data set first, and then you can apply it to a bigger data. Okay. So another type of alignment quality control or quality checking checkpoint will be this alignment quality check. So ClearMap produces a transformed version of the atlas image. What this means that once you do the alignment, you can convert your atlas image so that it is like on the same space as your raw image. So this is what's actually done. So the green on top is this MRI image provided from the Kim atlas. The red one is an actual raw image. So these two are already done, already went through this alignment process. So in an ideal world, world, these two should align perfectly to each other. The trace here is what we use as a metric. We have this F1 score, how well these two images align to each other. You can kind of appreciate that the brain sections in the middle, these ones have a very good alignment, while you start to see some things that are like fading off in the olfactory bulb or in the very, very hidden part. So somewhere here, somewhere around here. So this is what we typically observe when we do alignment in ClearMap. You see this very high F1 score in the middle, and then you see a very low one in the initial part and the very late part. It seems that the most anterior part and the posterior sections are very likely to get misaligned, especially when the olfactory bulb and the very hidden part very subjective to very like easy to damage and gets like very variable shapes and like structures. So the alignment of ClearMap is not like robust enough to like completely warp weird shaped olfactory bulbs to their original position. It's like it kind of leaves it leave it as it is. If you really want to be precise and want to like care about the microstructures in every single part of the brain, ClearMap is not necessarily the best option. You have to go into like using manual approaches like Sam mentioned in his first part of the talk. Another approach is just to omit those regions. So like say like you have a cutoff, like any region that is before this like Z position or like, yeah, AP axis, anything that's after this AP axis will not be quantified. You can use those kind of things to just like omit some of these less low alignment quality regions.
So you can imagine then like this is highly dependent on your dissection, your quality of your dissection. So if like you're missing a lot of your olfactory bulb, then you know the curve that he's showing at the top is going to be dramatically changed because of that. Likewise, like if the hindbrain, if you're missing a chunk, or if like you know you accidentally take a big chunk out of your cortex, that's gonna like probably affect the alignment. So it's just you need to be aware as you're doing your dissections. You know, take good notes so that when you get to this point and you are looking at the alignment, you can remember as to why it's giving you certain characteristics that it is, and then that can affect your downstream segmentation. So just to be aware.
If you're particularly sitting in more anterior or posterior region, would you want to then sort of adjust where you're centering when you're taking pictures and doing these sorts of things versus like the center proper anterior and posterior? Does that make sense?
So I think there's obviously the raw image, the tissue that you're handling with should be as close to your atlas. That's key part one. There are some more like robust type of alignment algorithms, so you can look into that. For like this is a more like a general thing, but there's not a good, I think, template atlas for like the spinal cord or anything posterior. So that's like a very completely new topic. You should just need a better atlas for that so that you can use it for alignment. Okay. Okay, so now we're gonna talk about actually running ClearMap preprocessing. I like to think about it as become a cooking situation. I like to cook, by the way. So ClearMap makes things like this. You have this, oh, this is not, no, this, you have this processing script, and then you have this parameter script. You can think of it as this your recipe book and your ingredient list. So all the ingredients, how much what, how much what, how much what will be listed in the parameter script. And then what to do with those will be listed in your pre-processing, in your processing script. If you think about like all the functions that we talked about, those will be listed in the processing script. So you will be saying that first you will do this, second you will be doing this, third you will be doing this, just as a recipe. And then for the parameter script, you'll be, okay, use this value for this step, use this value for this step, and use this value for this step, okay? So each data set will have their own processing script and parameter script. And then you will use that to process things. Okay. Okay, so obviously to make a dish, you need a kitchen. And in this world, that means like you need a computer. So, and also what you want to do is you want to have a really good computer. If you want to do this on your like laptop or whatever, you want to have good memory and good CPU cores. The number of CPUs or number of CPU threads really matters for the speed because ClearMap uses a thing called parallel processing. If you have like a big chunk of meat, it will take forever for you to cook. But if you can chop it in 4 and you have a 4-burner stove, you can cook it 4 times faster because you can just put it on 4 parts. That's the kind of concept of parallel processing. If you have a big chunk of images, you can chunk it into like quarters. You like do like z size 1 to z size 100 in this part, z size 101 to 200 in this part, and so on. ClearMap does it for you. So what you likely want is like a lot of memory, but also like CPU with multiple cores, so you can like allocate specific jobs for each of these memory cores. ClearMap unfortunately recommends Linux OS. This is also a hurdle for a lot of people. I don't think a lot of people use Linux usually, but Linux is like a lot more easier to like install ClearMap. Okay. The reason I recommend a high performance computer cluster is that it's actually really hard to make your kitchen. It also is very hard to change your kitchen once you make your one. So one day you might be like just having mouse brains at like 1.6x, but like maybe your boss will come to you and just ask you to just start analyzing 3.6x rat brains. And then you're like, that can't work on your like computer that you constructed. So, but if you work on a high performance computer cluster, it's typical that they will provide more like larger memory nodes or like more CPU, more nodes in general. So like, okay, so today we use this one or this data set using a more better node. So you can do things like that. Another thing you can think about is that if you have one computer, you can typically run one brain. So you have to sequentially all of your data. Again, if you are working on a high performance computer cluster, which typically provides multiple nodes, you can just assign 1 node per brain. And if you have 20 brains and 20 nodes, you can just assign everything in parallel. So it will speed things up like remarkably. And that's the reason I want to like, I typically recommend getting access to HPCs. Okay. So a computer cluster, what is a computer cluster? Typically, this is a kind of normal computer. You have your own computer, everything is in here. You have your CPU and you have your memory, everything is in here. A computer cluster is a bit different. So in a computer cluster, typically they provide a login node, which everybody goes into. That login node has no like powerful processing power, so you can't do anything on that. But what you do is that you say to this login node and to this system that, okay, I want to do this type of job. I want to use this kind of processing units to do this kind of job. And then the cluster side will provide you with compute nodes. So like, okay, so you want to do this. Okay, I will give you this node, and then you can get access to this node for this amount of time. So that's how like people share a lot of computer resources. That's why it's a bit different from your like local computer. You kind of share these compute nodes. You allocate jobs, what we allocate jobs per compute nodes per user, and then kind of use everything as like a group. Okay. Yeah. So what InStuberLab have been trying to started to use is to get access to computer clusters through a NSF program called Access. So as it says here, Access is a program established and funded by the NSF to help researchers and educators with or without supporting grants to utilize the nation's advanced computer systems and services at no cost. So this was really, really good for us. We actually purchased a node on the computer cluster in UW Hayek. This was, I think, $5,000 or something like that, or maybe more than 50,000. I forgot, but it was a good amount of money to just get one node. In the meantime, if you go get access through Access, it's not like you have your own node, but you get access to like a bunch of resources. What we will be using today is a HPC called Bridges 2 in University of Pittsburgh. they offer like 504 nodes for the regular memory types. They offer like 4 nodes for the extra memory type. The regular memory have a 256 gigabytes of RAM memory and 64 cores, which means that you can like parallel process like up to 64 things or up to 128 things. For the extreme memory, you have like 4 terabytes of memory and you have like 24 cores or like 96 threads. This means a lot. It's very hard to process a 3.6x brain with a normal memory. It takes longer, it's slower, and you can't even process things if it's like super big. It's nothing when you run things on extreme memory. So if you are planning on doing like projection studies in the rat brain, even projection studies in mouse brain, getting access to these types of nodes is critical, and the best way to do this is probably through this access program. Okay. I won't go through how to get your get a project in Access, but it's very simple, and you can just like question me when you start want to do this. Yeah, that's a good question. It's obviously depends on your computer. So like, I don't think it will be faster in your normal computer in the cluster. Like processing one brain on a cluster computer is not necessarily faster than on your like local computer environment. Transferring data is a lot more faster when it's like closed. But as I said, you can parallel that. So like processing 12 brains, processing 20 brains will be a lot more faster on the cluster computer. My like rough estimate is that you can process a 1.6x resolution mouse brain in the regular memory like within 1 hour or 2 hours. 3.6 hours will take longer. In extra memory, it will be like less than an hour, like 30 minutes to like hour-ish. And then again, you can like parallel process that. So like I process like 12 brains in like half a day or something like that. Okay, so now that if you have access to multiple nodes, now you want to start thinking about processing multiple data sets at once. So this is something like you're a master chef, you have this like master recipe and master ingredients. You want to allocate these recipes to like multiple chefs in multiple places. Our ClearMap has this function in it. So we call this like batch processing scripts. We have a list of scripts that you provide this process file for batch processing, this variable file, batch submit job script, and experiment parameter script. I will go into those in detail, but the general idea is that you have this Excel sheet which contains all the variables that you're going to use. And then you have this experiment parameters CSV file, which lists all the data sets that you want to process, and then like some variables specific to that data set. You submit those together with this process file and the batch submit jobs file, and then they will like automatically create job files for each of these data sets and submit those jobs to like to each node. So if you have 20 lists, 20 brains, and if you want to just process with like the same variables, you just like list that into your experiment file and then submit this batch submit jobs script, and then you get all of them, like all of the jobs necessary submitted to the computer cluster, and then you just wait. Okay. Okay, this is probably going to be the last thing in the morning, but another thing you need to think about is this environment issue. So as I said, like in order for ClearMap to work properly, you need a specific environment. And I think I talked about that as a kitchen. So you need a proper kitchen to make a specific dish. When you allocate jobs to like all sorts of nodes, you want to make sure that the same kitchen is recreated in specific nodes, right? So to do that, there's a framework called a container or a singularity container. The idea here is that you create a kitchen and pack it into one file, and then send the file to all of those like those small data sets and ask them to recreate the kitchen using that file. Okay, so in this case, the working environment for ClearMap is stored in a single environment file called this CIF file, and the processing script will be executed inside that container file. Okay, so you don't have to worry about installing some packages in a specific node. You don't have to worry about that. As long as you just provide the same environment file, you can recreate that environment as it was, and then you can execute the code inside that environment. It's a very useful way to process things or like run things in an HPC environment. But we already took care of that, so you just have to copy paste the zip file and then like you can get started working. Okay. This is some minor thing. ClearMap, it's actually a working version for Nape, is open on GitHub in my repo. So you can go there and check and like get some updates too. Some of the scripts, the template scripts that we will also be using today will also be uploaded there. The Singularity container, this is again, is a working version for Nape, is on this like container like sharing system called Scilabs. You can access this whenever you want. The Nape community also have some Lightsheet resources. The main thing will be this Google Drive. This stores some standard brain atlas files. It stores already trained elastic classifiers. It stores example of spot detection parameters. This will be better if more people start to use ClearMap. If people can provide their elastic files and then share it, it will be useful for more users. So I would encourage people to at least take a look at it and use it as a like a resource. We also have the Slack channel, Lightsheet and Clearing, community and Clearing, and also we also share resources there. There's also a monthly group meeting every last Friday on the month. So from 2 o'clock, every last Friday of the month, 2 to 3, and the next meeting is going to be the 21st. So everyone equals welcome. Okay. So here, that's all for the first part. And then after this, we want to go on to hands-on training. I want to ask people to kind of move because Mac people and Windows people don't get along with each other. But like Mac peoples, please sit on the left side of the room, and the Windows users, please sit on the right side of the room. And then there will be specific personnel you will be more familiar with your OS. Okay, thank you. It will be fine. If yeah, you can come on and watch. I will be doing things too.
Is everybody Windows?
Yeah.
Huh?
Windows over here.
Okay, somebody's Mac that I was there.
No Linux users are saying that. I don't know.
You need to go that way. That's because it's not about recently, so. I want you to listen.
Yeah.
I think it's
Wasn't there like that?
It actually makes
That's
sense
so-
for a
so
lot.
politics here.
Yeah, got like green stuff. I mean.
It became more,
Start making fun of the next guy.
it became more if you have a Windows, where you get to sit at the table.
Yeah.
You would be over there.
Maybe, yeah.
So given instructions beyond just cluster.
Or is it a good thing?
Oh, yeah. Great. Yeah, we can pass a break for
Just to get the Macs up and running.
Hello, Zoom people. We will be having a 15-minute break. We will get back in like 10:05.
Yeah, I don't do any. I usually use Windows and go to. Yeah, I don't like Chrome. Windows is way better. I have got this. It like uses up the whole screen, but I don't have to.
So Ken,
Mhm.
we haven't been given any other instructions so far beyond this like.
Yeah, it should be
next to
going
the
across
cluster.
browser.
Yeah. That's great. Like if you have already connected to the cluster, you're way ahead of most people. So that's great. Were you able to log in and change the password smoothly?
Yeah, so I already got like my password set up and. So we can use that. I got the term.
Yeah.
Oh, perfect. Yeah, let's see how it goes. Yeah. Hopefully, like hands-on part is always a bit worrying. Like you might get some random errors from random people.
This might be all set up for then, but they're gonna switch.
Yes.
Yeah.
That's some kind of-
Yeah, a lot of shorter.
Okay,
Or is that something I need to start? At some point.
I'll see if I can remember the instructions.
It's like I've never dealt with it, because I was like, so much of what I do, I have to be here, and then I was like, there's been one or two times where it's like.
Yeah.
There, I was sick or something, and so I wasn't doing it, I was just like.
He walked me through both ones, and I did manage to replicate it to get the lack of thought behavior. I could do stuff along. Yeah,
Right.
so yeah, I can replicate it, it'll take me a little bit of time.
And I just feel like.
But yes.
when I was doing rat stuff.
SCH.
It's an experiment with this. I did notice that we were. Stuff that we had.
I'm gonna forward you this. Think about it.
So I'd have to get mine, otherwise just. Yeah, it's cool. Okay.
Yeah.
The Windows one is perfect. I know, it only is put all in one room and get, like my own.
I remember, it's like Susan asked.
And everybody in it.
Oh.
Okay.
We get metal, I think.
It's, yeah,
Yes.
it's a lot better than.
Yeah, then those are things that you're doing.
So I think I started with it, I'm like, wow.
Yeah, I'm here today. Like, my life. So like, it's not like, but
It's not, that's a.
it's not like, it's a computer now, when I bought it, it was a gaming
And my.
computer because I was like,
Tax
gaming
time.
computers and stuff.
Set up.
Like, it's like a game,
Never happened.
bothering.
But if
It's like gaming computers, I figured out something on average to function if I have like script open and 50 tabs.
Stuff that,
Whereas
like, they'll turn
it's
around.
my,
and
it's
say,
my
"Yes,
non-effort way of ensuring that.
Sometimes it's turn around and they'll, like, they're kind of figure out instead of like the wall and stuff.
The issue I was
They
told,
use
so
their
it's gonna be so good, they
Yeah.
basically.
Bridge.
Yeah, mine is, it's like probably a
Don't have.
I think it's a smaller one.
Generally. Yeah.
Was
Right.
video
And that's
parts of
fair.
it in the.
I mean, you know, it's segment metal.
That makes sense to me, because if you're like a videographer.
Yeah,
Like it makes
yeah,
that.
processing in the field. Is like, yeah, this was several years ago, I did a request at some point.
Yeah. Let's speak about that.
Yes,
Magic the Gathering. It's like. Accessory graphics.
But I might just.
Like the Magic the Gathering arena does a lot of excessive graphics.
yeah.
Like it's actually.
It's a goddamn.
So, yeah.
You need to spice it up.
Yeah.
It's the most ridiculous amount of.
What's the idea of
Yeah.
Or what's the recommendation?
Which is, it's so funny that something back to crash, and meanwhile, you can also, like, I've seen the phone game, like, it's like, that should be running.
Yeah, I
on
was thinking that
phone,
too, and I think.
Right.
I think that.
No, meanwhile, it handled like. The same computer is just fine with my concept.
Yeah.
And can handle 50 different like tabs and internet access. But yes, it's a card game crash.
Yeah.
But.
But yeah, I will say that, having the cluster on your laptop is really nice, because basically, like, when I started here, I was worried about the desktop, but now it's like, basically, I have like less and less stuff on my laptop now, and like, And as well as my, like, request on, like, I basically just like, I just like, I'm just gonna log on to the desktop, because everything.
Yeah, yeah, I don't know why I was working on phone. The one time, a couple days.
Right, I don't know, maybe. But you can even end up with a weird switch where it's like, well, it didn't break.
I assume it was when the.
15%. Somehow things will fall into place, yeah. Yeah.
But yeah, it was very much a like, okay, what things can I find the genetic files online?